Good morning, students. I am Mayank Tiwari. Today we will solve test number 58 of the General Agriculture series. Let's start. Student question number 1. Hybrid of cytoplasmic organelle is called. You have to tell what is this hybrid is known as. If we produce a hybrid of cytoplasmic organelle, what is this known as? Option A, hybrid. Option B, symmetric hybrid. Option C, asymmetric hybrid. Option D, all. So student, first we discuss symmetric hybrid. Symmetric hybrid is what? When normal protoplast of two species are fused. It is called symmetric hybrid. Please remember it. Example of symmetric hybrid, student, is pomato. Pomato is cross between potato and tomato. This I told you, pomato is produced by Dr. Ashish Datta. So please, sorry student, pomato is not produced by, I think it is potato. Potato, it is produced by Dr. Ashish Datta, not pomato. Pomato is a cross between potato and tomato. So please student, remember potato, it is produced by Ashish Datta. I will just check what is the difference between potato and this pomato. And student, some of the application of somatic hybrid are production of hybrid between non-flowering or a sterile plant. If you want to produce a hybrid between non-flowering or a sterile plant, you can use somatic hybrid. It widens the this genetic base, like wheat into rye, you get critical. So it is a somatic hybrid. Student, next is asymmetric hybrid. In asymmetric hybrid, we exclude the genetic component of one species and include the genetic component of one species. It is asymmetric hybrid. Please remember asymmetric hybrid, student. And hybrid is most important. Hybrid is hybrid of cytoplasmic organelle. The question I asked. In hybrid, we cross enucleated cell into nucleated cell. Please remember it. And hybrid, student, please remember it is a hybrid of cytoplasmic organelle. In hybrid, we fuse protoplasm of one parent and cytoplasm of another parent. So please remember hybrid is also called cytoplasmic hybrid. Student, question number 2, you see antibody produced in plant used to cure many diseases. You have to tell what is this antibody known as, which is produced in plant and it is used to cure many diseases. Option A, transgenic. Option B, cisgenic. Option C, intragenic. Option D, plantibody. So student, all the options I am telling you. Transgenic, you might be knowing, student, gene transfer between unrelated organism. It is called transgenic. Example of transgenic you all might be knowing, it is BT cotton. First transgenic plant was tobacco. Please student, remember that first transgenic plant was not cotton, it is tobacco, and it expressed caniamycin resistant gene. And student, what is cisgenic? Cisgenic means natural gene coding for an agricultural trait. Source of gene is related plant. So please student, remember in a transgenic, source of gene is not related organism, but in cisgenic the source of gene is related plant. What is intragenic? Intragenic means technology that insert gene cassette from a specific sequence of crop that belong to the same breeding pool. Please student, remember intragenic also. Intragenic means technique that insert gene cassette containing a specific genetic sequence from crop that are belonging to same genetic pool. So student, please remember these terms, transgenic, cisgenic and intragenic. So the correct answer of this question, student, was plantibody. Plantibody are the antibodies produced in plant. Student, please also remember one term, molecular farming. What is molecular farming, student? Utilization of transgenic plant to produce specific chemicals or pharmaceuticals, immunoglobulin, polyhydroxybutyrate. It is called molecular farming. So please student, remember this. Student, question number 3, you see Pokali is an indigenous cultivar of option A, rice. Option B, mustard. Option C, sugarcane. Option D, lathyrus. So student, you have to tell what is this Pokali, what to which crop this indigenous cultivar belongs to. So student, this question can be important for you. This salt tolerance, a variety of rice developed for salt tolerance, BTS24. It has been developed from Pokali rice cultivar, and it is a soma clonal variety. Please student, remember BTS24, it is a soma clonal variety and it is developed from indigenous rice cultivar Pokali. So Pokali is the right answer of this question and it is an indigenous cultivar of rice. Now students, soma clonal variation, it is often asked what is it? The variability generated by use of tissue culture technique, it is called soma clonal variation. And who gave the term soma clonal variation? It is also asked. Larkin and Scowcroft, they gave the term, they coined the term soma clonal variation. Please student, remember it. Student, some application of soma clonal variation, they are often asked in objective question, like improved geranium variety velvet rose has been developed by soma clonal variation. Pusa jackson variety of mustard, it is developed by soma clonal variation. CMAP or bio 13, it is a variety of citronella, it is also developed by soma clonal variation. In maize, we have developed glyphosate-resistant character by somaclonal variation. In wheat, grain color and protein content. In soybean, protein plus oil content. Brassica knapus, glucosinolate content. Sugarcane disease resistance. And student, one thing you remember, for cold tolerance, we increase the proline content in tissues. For cold hardening, we do it by somaclonal variation. Please student remember it. For cold hardening, we increase the proline content by somaclonal variation. And student, one thing also remember that this lathyrus contain neurotoxin. So first variety of lathyrus having low neurotoxin, BioL212, it is developed by Mehta and Santha. So please remember Mehta and Santha developed a variety of lathyrus BioL212 for Central India zone, which is having low neurotoxin. Please student remember it. Student, now see a very basic question. Optimum temperature for Rabi crop. Option A 20 degrees centigrade, option B 30 degrees centigrade, option C 25 degrees centigrade, option D none. You have to tell what is the optimum temperature for Rabi crop. Student, in this question many of the student confused, so please remember, if somebody asks you optimum temperature for any of the Rabi crop, it is 20 degrees centigrade. For any of the Kharif crop, it is 30 degrees centigrade. For any day neutral plant, the optimum temperature is 25 degrees centigrade. So student, please remember the optimum temperature of Rabi, Kharif and day neutral plant 20, 30, 25. And student, also please remember this National Seed Research and Training Center, it is located at Varanasi. Please student remember it, National Seed Research and Training Center, Varanasi. And in Varanasi is also Indian Institute of Vegetable Research, IIVR. Student, while you are reading seed science, please also remember tetrazolium test. Tetrazolium test is given by Lacon. Please student remember, Lakon, Lacon gave the tetrazolium test. The red color development is due to formazole. Please student remember, it is often asked what is the cause of red color development in tetrazolium test. It is formazole. Once you stain dicot cotyledon with tetrazolium, the whole part of dicot seed is living, so whole seed will be colorful. But in case of monocot seed, only the embryo is living, so we have to dissect the seed first and then stain. So only embryo is living in the case of monocot seeds, so only embryo gives color. But in dicot cotyledon, in dicot seed, all of the seed give color. Student, question number 5 you see, it is very easy question. DCMU inhibits option A cyclic photophosphorylation, option B non-cyclic photophosphorylation, option C Hatch and Slack pathway, option D Kelvin cycle. So you have to tell what does DCMU inhibit. So student, please remember light reaction involve two type of photophosphorylation. One is non-cyclic photophosphorylation, second is cyclic photophosphorylation. What is non-cyclic photophosphorylation? When photoexcitation of chlorophyll molecule occur, electrons start, the evolved electron after passing through ETC does not return back to the same chlorophyll molecule. It is ultimately trapped by NADP, which get reduced to NADPH2. And in case of cyclic photophosphorylation, excited electron from chlorophyll after passing through ETC return back to the same chlorophyll molecule. It is cyclic photophosphorylation. Please student remember cyclic and non-cyclic. In cyclic, excited electron from chlorophyll, once it passed through ETC, it will come back to the same chlorophyll molecule. But in non-cyclic photophosphorylation, excited electron after passing through ETC does not return back to the same chlorophyll molecule. That is why it is called non-cyclic photophosphorylation. Some more points students you remember about non-cyclic and cyclic. In non-cyclic photophosphorylation, both the assimilatory power are formed, ATP and NADPH2. Both are formed in non-cyclic photophosphorylation, but in cyclic photophosphorylation only one assimilatory power is formed, that is ATP. In non-cyclic photophosphorylation, both the photosystem 2 and photosystem 1 are involved, but in cyclic photophosphorylation only photosystem 1 is involved. And the splitting of water molecule occur in pH2. Therefore, evolution of oxygen take place in non-cyclic photophosphorylation. No splitting of water molecule, no oxygen evolution. And student, this non-cyclic photophosphorylation is stopped by use of DCMU. DCMU is your dichlorophenol dimethyl urea. Dichlorophenyl dimethyl urea, it is a diuron, it is a VD-side. So this stops pH2 for non-cyclic photophosphorylation. Please student remember it. And cyclic photophosphorylation is not inhibited by DCMU. And this non-cyclic photophosphorylation occurs in all the classes of algae, bryophyte, pteridophyte, gymnosperm, angiosperm. And this cyclic photophosphorylation occur in photosynthetic bacteria, chlorobium, chromotium, and rhodospirillum. Please students remember these two. These are very important. Student, now some important difference between C3 plant and C4 plant. Student, also remember that C4 cycle is also called Hatch and Slack pathway, and C3 cycle is also called Kelvin cycle. In C3 cycle only one type of cell are involved, that is mesophyll cell. In C4 plant Both the cell are involved mesophyll and bundle seed cell. C3 plant, no Kranz anatomy. Kranz is a German word which means red. And in C4 plant, which shows C4 cycle H and slack pathway, Kranz anatomy is found in the leaf. In C3 plant, only one type of chloroplast is found, that is granule. In C4 plant, both the type of chloroplasts are involved, agranule and granule. In C3 plant, first CO2 acceptor is rubisco. In C4 plant, first CO2 acceptor is papcase. In C3 plant, first CO2 fixation product is 3-PGA. In C4 plant, first CO2 fixation project is oxaloacetic acid. Student, C3 plant, only one carboxylase enzyme involved, that is rubisco. In C4 plant, both the carboxylase enzyme are involved, rubisco and papcase. In C3 plant, CO2 fixation rate is less. In C4 plant, CO2 fixation rate is more. In C3 plant, less energy is required. In C4 plant, more energy expensive they are. And inhibition of photosynthesis by oxygen in C3 plant, no inhibition of photosynthesis by oxygen in C4 plant. Photorespiration is high in C3 plant, no photorespiration is found in C4 plant. C3 plant productivity is less, in C4 plant productivity is more. High CO2 compensation point 25 to 100 ppm. And this C4 plant have low CO2 compensation point. Student, this is asked what is the CO2 compensation point of C4 plant. So please remember 0 to 10 ppm is the CO2 compensation point for C4 plant, while in this C3 plant have a CO2 compensation point is 25 to 100 ppm. Optimum temperature tolerance range for C3 plant is 20 to 25 degrees centigrade. Optimum temperature tolerance for C4 plant is 30 to 45 degrees centigrade. And C3 plant example, rice, wheat, potato, chlorella. And C3 cycle was first discovered in chlorella only. And C4 plant example is all the member of Gramineae except wheat and rice. Wheat and rice are C3 plant. And amaranthus, these are example of C4 plant. So student, that's all for today. Thank you.